Good morning everyone. I am so glad again to meet you all. The same chapter, final episode of chapter 18, Heredity. I hope you all are going through the lesson keenly and concept by concept, page by page, not just reading at the last moment or not reading only the question answers. Only if you read concept and concept by concept or page by page, I don't think whatever you learn it's for your exam alone, for your test alone, for your mark alone. It is for your life. Okay, so it's not, I don't say based on your job or based on your future. Apart from that, whatever you learn, again I say it's not only science, it may be English or Tamil or maths or social. So if you keep on learning something, you will be something special in your future. And not only that, since we learn heredity, when you are intelligent, surely your next generation will be or supposed to be intelligent. So also keep that in mind. If you skip, your offsprings or your children will also skip many. Okay, so usually they say if you are skipping 8 times or if you are doing something 8 times, our kids will be doing double the times. Okay, so keep that in mind. Either whatever you do positively or negatively, our kids will be doing twice, sometimes even thrice, because when we look at present new generations, you find those kids they are more studious, they are more intelligent, and they are more advanced than we people. Compared to my generation, you will be surely advanced, and compared to you people, your other generations, so who are all in the kid stage, very young stage, they will be more advanced. You should have noticed by this time. So keeping this in mind, try to read all the concepts, all the pages of your subjects. This can be extended to some extent, where you can also go through other books, or nowadays we have this internet, where you can learn many things apart from what is in the book. So when you have doubt time being, you may not be able to ask your particular teacher. So with your parental guidance, so try to Google up certain concepts, learn certain things, so that it is easy for you to understand and recall. Okay, so again I say it is not only for your exam, it is for you. Okay, so keep that in mind, and let us enter into today's chapter 18, Heredity. So we are at the last part of our lesson. So today we will end this lesson with the topic mutation. Okay, so mutation. So this is a little complicated concept, at the same time easy to understand. Okay, that is actually the definition is easy, but the process is actually complicated. Okay, so sometimes it is advantageous and sometimes or mostly it is disadvantageous, harmful. Okay, and it may result in sometimes even death. So mutation is very, very dangerous. So like how we should not be careless with certain things, like that also don't be careless with mutation. Okay, so when it comes to this mutation, actually what is mutation? It is an inheritable sudden change in the genetic material of an organism. That is actually called as mutation. So simple words I say, it is the sudden change in the genetic material, that is DNA, okay, or chromosome. It may be as per actually in your book it is given DNA, but it may be in DNA or in the chromosome, but a sudden change. That sudden change which is inheritable. Unless it is inheritable, you cannot call it as mutation. Okay, so when I say mutation, for example, we know how a human body will look like, Suddenly I have a horn, okay, or I have a long tail, like ear like a cow or a buffalo or any cattle. Okay, so that is actually mutation. But only thing is that even though I have a sudden change, only if it is inheritable, then only it is called as mutation. Unless then it cannot be called as mutation, because there are some people in this world who have a horn. Okay, so I don't know whether you believe this or not. There are some people who have horn, but I don't think they are inheritable. I hope you know what is inheritable, where this character or this DNA which is responsible for that particular character is passed from one generation to the other. So that is important. So inheritable sudden change in the genetic material is actually called as mutation. Okay, so this word you have learned in various places, especially in movies. You come across this word mutant, mutagen and mutation. Okay, so mutation is the process where there is a change. Okay, one, mutation. Second one is the agent that causes mutation are called as mutagens. Okay, and those who have a mutation in them, okay, maybe it may be human or animal or plant, those people or those organisms are called as mutants. I hope this word you come across in certain movies like X-Men or some other. Okay, so I would say wherever when you come across this superficial or what to say, some extra power, okay, which is against nature or which is above nature. Okay, nature in the sense above a normal human being. So surely that the power or that character is because of mutation. So I hope this will help you to understand what is meant by mutation. At the same time, so usually in movies we come across a positive mutation where a man gets the power of a spider, or he resembles a what to say, characteristics of all many living organisms. That is characteristics of chameleon, characteristics of lizard or reptiles, so put together in one particular person. So like that, in movies you come across mostly positive reaction of mutation. But I would say, that's why I said be careful with mutation. That is, mutation results in certain problems. For example, cancer. So mutation, the main reason for cancer is mutation. Okay, so that's why I said be careful with this mutagen or mutant or mutation. Okay, so don't expect that you will get some superpower and go and stand before a gamma ray or a alpha ray or some other X-rays. Okay, so all are problematic. So be careful with this agents that causing mutation. Okay, so before going to the next concept, so something I just want to say, that is, when it comes to mutation, there you can classify the agents that causing mutation into three types. That is physical, chemical and biological. So you have certain physical agents which causes mutation, you have some certain chemical agents that cause mutations, and you have certain biological substances that causes mutation. So which is not in a book, just have in mind. Okay, physical agents which include rays, just now I said. So actually when it comes to light, you come across light can be divided into two spectrum: visible spectrum and invisible spectrum. Okay, so visible spectrum, what we are about to experience, okay, which includes VIBGYOR, that is VIBGR, violet, indigo, blue, green, yellow, orange, red. So which is safer, and you call this as visible spectrum. Whereas in invisible spectrum, where actually you come across a certain rays, harmful rays, which is not visible to our eyes. That's why they are called as invisible or non-visible. I think correct word is invisible. Okay, invisible spectrum. So which include X-rays, gamma rays, alpha ray, beta ray, gamma ray, UV, ultraviolet rays, IR, infrared rays. So all those comes under a particular wavelength, and so we categorize them as invisible spectrum, and those rays are mostly harmful and they are mutagenic in nature. That is, if you come across these rays, if these rays if you are exposed to these rays, surely that results in mutation. Okay, so be careful with these rays. Okay, so time being, I think you should be knowing even sun has these rays. Okay, since we have this ozone hole, we people, because of causing pollution, we openly call all the rays to come inside our atmosphere. Okay, so I think you know about those factors, ozone layer and where you come across ozone hole, and through this ozone hole all the harmful rays that are coming inside. Okay, right. We'll stick on to mutation, so I'm not going into ozone or ozone hole. Okay, so these are called as physical mutagenic agents. Okay, so alpha, beta, gamma, then you have UR, IV, okay, then X-ray, so all these are harmful. So don't be lethargic with these rays. You should be very careful with these rays, okay, because they cause mutation. Mutation, again I repeat, don't think just horn growing from your head, or maybe you are getting gigantic like a Hulk, or you are able to produce a web like a Spider-Man. Okay, it's not like that. Basically, I would say only one concept. There are many harmful effects of mutation. Basically, it results in cancer. So be careful with rays. Okay. Then second comes the chemical. That is, there are certain chemicals. Whether you know or not, these chemicals, they are even added to this certain preserved foods, canned foods or tinned foods. So be careful with these foods, because as a preservative they add certain chemicals, and those chemicals, they are mutagenic in nature. So be aware of that. Okay. Similarly, one more thing I left, that is the smoked foods. I hope you know what is in the smoked foods. In hotels you come across certain foods that are smoked in coal. Okay, so even that also can cause mutation. So avoid more smoked foods. SMOK, okay, smoked foods. Okay, find out what is under smoke. Okay, so don't rely on me. Okay, then finally there are certain viruses which causes mutation. So if you look into the reproductive way of viruses, so viruses, what they do is that they enter into a cell and they reproduce. That is the nature of viruses. That's why they are called as intracellular parasite. Okay, so usually bacteria or fungus, they stay away from the cell. Even though they enter into the body, they don't enter into the cell. They stay away from the cell and cause infection, either by producing toxin or doing harm to our cell. Okay, but they will be staying outside the cell. But these viruses, all viruses, they cause infection by entering into the cell, and so they incorporate. I will even add to this, okay, so that you will understand. They will incorporate their genetic material into our genetic material, and they undergo reproduction. So as we learn this DNA replication, they initiate our DNA to in such a way that their DNA also reproduced or replicated. Because viruses, you know, they are called as non-living substances. Okay, so they cannot do any function of their own. So they deal with a host cell. Only with the help of host cell they undergo all the process. So in that way, viruses, they are mutagenic in nature. They come under biological agents. So I just said only few. So physical, chemical and biological. There are three types of mutagenic agents. Okay, then mutation. There was a scientist named Hugo de Vries in 1901 who first introduced this term mutation. Okay, Hugo de Vries. Okay, he observed the phenotypic changes in a plant named as evening primrose. The scientific name of this evening primrose is Oenothera lamarckiana. Okay, so he identified certain changes in that plant, and based on that only he named the term mutation. Okay, now coming to the main core, types of mutation. There are two types of mutation. One is chromosomal mutation and second one is gene or point mutation. Okay, so by this time you know what is a chromosome, what is found in the chromosome. Okay, fine. So I don't want to go into that. So if a mutation or if a change takes place in the whole chromosome, somewhere in the chromosome, okay, not the whole, somewhere in the chromosome, then it is called as chromosomal mutation. And if the mutation takes place in a nucleotide of a DNA, I hope you know, now you understand what is meant by, okay, or in a particular gene, somewhere in a gene, then it is called as gene or point mutation. I think this is very easy. Okay, if the mutation takes place in chromosome, okay, the whole chromosome, somewhere in the chromosome, then it is called as chromosomal mutation. If the mutation takes place inside, that is inside the chromosome, you have DNA. In the DNA you have genes, and genes made up of nucleotides. So if there is a mutation in the DNA, especially in the nucleotide, one nucleotide or a sequence of nucleotide, then it is called as gene or point mutation. So let us first learn about this chromosomal mutation. This chromosomal mutation again can be divided into two types as change in structure and change in number. If there is a change in structure of the chromosome, it is called as, which is categorized as change in structure. If there is a change in a number, it is called as categorized as change in number. And this change in number can also be called as ploidy. Okay, we are going to learn later ploidy. What is ploidy? Okay, because there are two types of ploidy, we are going to learn later, euploidy and aneuploidy. Okay, right. So now let us start with this change in structure. That is, the change in structure may be by deletion or duplication or inversion or translocation. I hope you know what is meant by deletion. That is, if this is a chromosome, a particular part being deleted in a chromosome, if a particular part is deleted, it is called as deletion. Okay, so the chromosomal mutation, that is in structure, it may be caused due to deletion. Okay, I hope you understand what is meant by deletion. Then second, again the chromosome. Okay, so you have a chromosome. So in that chromosome, where you come across a particular sequence duplicated, that is, a sequence like this is added here, duplicate, okay, a copy of that particular sequence. So for example, if it is like this, so again you come across a sequence like this. So what happens? The chromosome, actually the length of the chromosome becomes long because of the duplication. Duplication is making another copy. So already you have a set. So like that another set is formed. That is actually called as duplication. So again I repeat, the change in structure may be by deletion or duplication. Then third case, it is called as inversion. Okay. So what is inversion? Where, so you have a chromosome. Here you have like this. Okay, this segment. So if it is like this, okay, so here I find more lines. So just to make you understand, I'm trying. So what is inversion? Where the chromosome will be like this, where this part will come here and this part will go there. So that is called as inversion. So you come across more lines here. So just for understanding, I'm saying it's not, there is no lines at all. Okay, so that is called as inversion. That is, for example, if this is like this, in inversion you come across chromosome like this. So it is inverted. A part of a chromosome is inverted. That is called as inversion. Deletion, duplication, inversion, and the fourth one is translocation. What is translocation? Again, if this is the chromosome, okay, so everything we are learning in chromosome level. Okay, so where for example this part, okay, so let me draw like this, this one translocated somewhere else, so here it is not present. Okay, so that kind of process is called as translocation. In all these three ways you come across chromosomal mutation based on structure. Okay, so one, where a particular chromosome is deleted, a particular segment is duplicated, where a particular segment is inverted, and lastly it is translocated somewhere else. So these are the four different kinds of change in structure. Okay, deletion, duplication, inversion, and the translocation. Okay, with regard to change in number, you have two types. One is addition, second one is deletion. So already we have learned about deletion. So addition, where a new set of chromosome is added here. So what happens? It is added here, so it becomes long. Okay, so in this case where the segment is deleted, okay, here the segment is added, so you come across a change in number. Okay, so like that you have two types of change in number. We call it as ploidy, so it may be either by addition or by deletion. Okay, now this change in number, that is ploidy, that can be classified into two types as euploidy, aneuploidy. So you know very well, already we have learned about this euploidy. Eu means true, eukaryote, okay, eucoelomate, like that we have learned this U in various cases. So U means true, okay. So euploidy and aneuploidy. So in euploidy, where the individuals have more than the usual number of chromosome. I hope you know what is the usual number of chromosome. The usual number of chromosome is 2n, that is diploid. When an individual has a mutation where if there is more than the usual, that is, I hope you know how you got this 2n diploid. This is natural condition 2n. It is obtained by a set of chromosomes. We have learned before, chromosomes always will be found in pairs. So with this, if another n is added, then it becomes 3n. To this, if another n is added, then it becomes 4n. So that is actually euploidy. That is, already you have a pair of chromosomes, a set of chromosomes. You call this as 2n, and this condition you call this diploid. To this diploid condition, if another set of chromosome is added, then the chromosome becomes a triploid. If to this triploid one more set is added, it becomes tetraploid. Tetra means 4. Tri means 3, di means 1. Okay, so if only one is found, it is called as a haploid or monoploid. Okay, so pair is present, so it is called as diploid. To this, if one set of chromosome added, it becomes 2n plus n, 3n. To this, if one more is added, then it is called as 4n. And also have in mind, it is not that only to diploid, to triploid n is added. Sometimes to the diploid you come across addition of 2n, that also becomes 4n. Okay, so it is not that triploid becomes a tetraploid, always it starts from diploid. This is the natural condition. So diploid, if 2n, another sets of another pair of chromosomes are added, it becomes 4n. Okay, so like that you come across two conditions in this euploid. One is triploidy and second one is tetraploidy. If it is triploid in nature, it is called as triploidy, that is 3n. If it is a tetra condition, then it is called as tetraploidy, 4n, 3n and 4n. And in triploid, in this case, mostly the plant or animal is sterile. I hope you know what is under sterile, where they cannot reproduce. Or in other way I can say seeds are absent. Okay, so in plant seeds are absent, so that condition is called as triploid. And tetraploidy, this is advantageous, not like triploid. Triploid is harmful because the organisms are sterile, but in tetraploid it is advantageous, where you come across increased size in the fruit and the flower if the plant is tetraploid. Okay, so these are the two conditions where you come across under euploidy, triploid and tetraploidy. Okay, triploid 3n, tetraploid 4n. Okay, so be careful, so don't confuse with aneuploid. Aneuploidy, where you come across only one or two chromosome. Okay, but here it is not so. A set of chromosome, n is a set, like how you come across in set language. Okay, n. Okay, so you come across a, b. Okay, you call them a set, like that. Here n is a set, to that another n is added. So this is a normal condition, 2n diploid, found in almost all the living organisms. So with this natural condition, if another n plus n, already you have 2n, with that 2n another n is added, that is called as 3n, that is triploid. And again with this 2n, if 2 more n, okay, that is 2n plus 2n, that results in 4n, that is tetraploidy. Again I repeat, triploid is to some extent it is good, because you come across seedless fruits, but the problem is with the animals, because the animals will be sterile. And in tetraploidy it is advantageous because the fruits and the flowers are bigger in size. Okay, now let us move to the next topic, that is aneuploid. So far we learned about euploidy, where keep that in mind, n we are learning about n. And in aneuploidy, here it may be, so actually here you come across always gain of a set of chromosome. Okay, but in aneuploidy it is not so. It is not a set, It is just number. Okay, so where you come across loss or gain of one or more chromosome. That is, again let me start with the 2n. So this is normal condition. To this, one chromosome may be added, or one chromosome may be deleted or removed, or in some cases you come across two chromosomes, or I would say absence of two chromosomes. So three conditions you come across in aneuploid. If there is one chromosome removed, that is you call it as 2n minus 1, this condition is called as monosomy. In this case, where you come across 2n plus 1, this condition is called as trisomy. So this one is called as monosomy, this one is called as trisomy, and here where you come across 2n minus 2, this condition is called as nullisomy. So there we use triploidy and tetraploidy. So ploid, where ploid means actually sickness. Okay, sickness by 3n or sickness by 4 sets of chromosomes. But here you come across three conditions: plus 1, minus 1 or minus 2. If 1 is added, so it is called as trisomy. If one chromosome is absent, it is called as monosomy. In this case, two chromosomes are lagging, and that condition is called as nullisomy. Okay, right. So we'll go to the next concept, that is Down's syndrome, which is a best example of aneuploid. Okay, so aneuploidy just now we learned. Okay, so under aneuploidy, where you come across monosomy, trisomy and nullisomy. Okay, so in this Down syndrome, actually what happens is the 21st chromosome will have another extra chromosome. An extra copy of chromosome will be found in the 21st. Okay, so if you look into your textbook, you find across an ideogram. Okay, already we have learned about ideogram. So you just refer 21st chromosome, where instead of two sets of, a pair, you come across three. That is that condition is called as trisomy. Okay, trisomy 21. Why it was called as 21? Because the problem is in 21st chromosome. I hope you know humans have 23 pairs of chromosome, and the last one is which decides your gender, male or female. Okay, so 23, 22, 21. So that 21st chromosome, instead of 2, or I would say instead of a pair, will come across 3. So that is this condition called as trisomy, Down's syndrome, which is named after the one who first identified it in 1866, that is Dr. John Langdon Down. So named after him, it was called as Down's syndrome. Okay, so don't think Down mean something down. Okay, it's not like that. The name of that particular person is Down, Langdon Down, and so it is called as Down's syndrome. I hope you know what is the syndrome. A collection of symptoms is called as syndrome. In AIDS you come across this word, acquired immunodeficiency syndrome. What is syndrome? A collection of symptoms. I hope you know what is the symptoms. Okay, right. So there are various symptoms found in this Down, that is, let me use the word trisomy 21. Okay, instead of Down syndrome, let me use the word trisomy 21. Okay, so there are various symptoms found in this trisomy 21. Basically, so if you look into this photo, you can understand. Even in book you have a small boy's photo. Basically these kids face a mental retardation. That is, their mental ability stops. It doesn't proceed, it doesn't grow after that particular period. Okay, that is in the kid stage itself. Okay, so mental retardation is found in these kids, this trisomy 21. Second one is a delayed development. So the process, the developmental process, generally physical, mental, all the process will be slowed down. Okay, delayed. Then third one is you come across certain behavioral problems. They won't be normal, they will be slightly abnormal. Okay, if you look into the photos, you can understand these kinds of kids you come across in our day-to-day life. Okay, then fourth one is weak muscle tone. That is, the muscles will not be strong, they will be weak. And lastly, these kids or these particular persons, they have a visual problem and hearing problems. That is, they cannot hear or visualize properly. So these are the various symptoms of Down syndrome. Okay, again I repeat, Down syndrome is otherwise called as trisomy 21 because the change occurs in the 21st chromosome of a human being. That's why it is called as trisomy 21. Okay. What is the change? An extra chromosome is present instead of a pair. Third chromosome is present, and that's why it is called as a trisomy 21. Okay, tri means 3. Already we have learned. Right. So I have sent the various symptoms. So go through the symptoms, and when you are asked a question like this, try to write like this in points. Okay, so that it is the presentation will be good. Keep that in mind, and I will continue. Okay. Now we'll go to the second type of mutation, that is gene or point mutation. Okay. First mutation. So far we learned this chromosome mutation, change in structure and change in number. In change in number, we learned two ploidy, that is euploidy and aneuploidy. Okay. So now we'll move to the gene or point mutation. As I said before, where the mutation takes place in a particular gene, in a particular nucleotide or a sequence of nucleotides. So that is called as gene or point mutation. In this gene or point mutation, it may be I said nucleotide, so it need not to be nucleotide, it may be even in the base. Okay, so a nucleotide comprises of a nucleotide comprises of a sugar, base and phosphate. So sugar always it is there is no problem with sugar, because in DNA fully you have a deoxyribose, and the phosphate also you don't have any problem, but the problem is the sequence of the bases. I said before, okay, the sequence A, G, T, C, 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 A, like that, each person will have a particular sequence. Based on that only we are all different from each other. That makes the difference. I have a skin color, my friend has a skin color, I have a particular height, my friend has a particular height. Even I don't need to go to my friend. In my family we have differences, my father, my mother, myself, my brother, my sister. So that differences you come across because of the sequence. At the same time, even though there are differences, you have a certain similarities that makes us together as one species, that is human being. Okay, so that you also keep in mind. Okay, so you have two things. One is you have differences, we learned in the beginning of this lesson, that is variation, and second one is we have similarities. Everything is based on this basis or base pairs, A, T, G, C. Okay, A binds with T, G binds with C. Based on that you have the DNA sequence, and based on that you come across various changes. Okay, right. And this gene or point mutation, it results in the abnormal protein formation. And that abnormal protein formation, it may be your hormones or it may be enzyme, and based on that your character is disturbed. Okay, so there is a change in your character. Okay, right. So this gene or point mutation can be classified into 4 types as substitution, deletion, insertion, inversion. So it is not in a chromosomal level, but it is in a base pair level, where when you come across A, G, T, C, something like so. Let me draw a sequence. Okay, where you come across A, T, G, C, 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 G, A. So like that you have a sequence. So by this time you should be knowing when A is on one side, what will be on the other side? T will be found. If G is on one side, C will be there. If C is there, G will be there. C, G, C, G, A, T, G, C. So like that you have this base pairs. So first is substitution, that is in one place, instead of, for example, let me choose this place. So instead of CG, if AT is there, if one base changes, automatically the other also changes. If A changes, automatically what will be here? T will be there. That base, the previous base will be lost. Okay, so because only there will be bind hydrogen bond between these two, not with the A and C or A and G. Okay, so if there is a substituted instead of GC, now AT is there. This is called as substitution. Okay, so this is in base pair level or nucleotide level. That also keep in mind. This before we learned in the chromosome level. Okay, and this kind of substitution is called is the first type. Okay, substitution. Then second, the gene or point mutation, where you come across one base pair or 2 or 3 base pairs deleted. This is called as deletion. Okay, second type. And third one is, as we discussed before, inversion. So before I had AT. If it is inverted like this, that is TA instead of AT. So A on one side, T on the other side. If it is inverted as TA or it may be of like this also. If these three bases, for example, I have let it be TGA, ACG. Okay, so it was inverted like this. That is A. That is like this. Okay, so A comes here. Already G is in the center, so it may be 4 or 5. Okay, so T comes here. This is also called as inversion. So if it is A, then T here, G, C here, then here A. So this is actually called as inversion. Okay, or insertion. This is inversion, and the next one is insertion. So I've changed the order. Okay, so no problem. So this is called as inversion, where the bases have inverted from one place to another. So it need not to be 3, it may be 5 or 7. So if there is full inversion, that is called as inversion. And last one is insertion. Imagine the same strand, extra 2 or 3 bases come. So here A, C. So for this you come across G and T. So which is actually, which does not belongs to this. A new base is inserted. Okay, so like that also mutation takes place. Okay, so it may be by substitution. Instead of one base pair, some other base pair may come inside. Second, particular base pairs removed, called as deletion. And third one is inserted. Okay, so a new base pair is inserted. 1 or 2 or 3 base pairs inserted, called as insertion. And fourth one is inversion, where the bases are inverted. Okay, so like upside down, that is called as inversion. So these are the 4 ways by which gene or point mutation takes place. Then an important type of gene or point mutation called as sickle cell anemia. I hope you know what is in the sickle. Okay, so this is called as sickle. Instrument used to for harvesting crops. Usually you come across red blood cells which are round, biconcave in nature, but due to mutation, that is gene or point mutation, what happens, the RBCs are made up of hemoglobin. So there is a gene, particular gene that is responsible for this hemoglobin. So when the gene, when that particular gene for hemoglobin is disturbed, what happens, automatically the protein, or the that is, I mean, I'm talking about hemoglobin, that hemoglobin protein will not be properly produced, and because of that, that results in the deformed red blood cells. As you could look into the slide, okay, you come across deformed red blood cells which look like a sickle. Sickle is an instrument, okay, to cut the crops. So there you come across deformation. I hope you know what is under deformation. Okay, change in the structure, and that condition, it is called as sickle cell anemia. Since red blood cells are affected, it is called as anemia. The cells appear like sickle, and so it is called as sickle cell anemia. So this is a important example of gene or point mutation. Okay, sickle cell anemia. The basic concept is, you know very well, everything that you come across in your body is controlled by a gene. So that gene, I said before, actually a DNA. In that DNA you have gene. Genes produce RNAs. RNAs produce proteins or some other chemicals that controls your character. Okay, so if that DNA or gene is affected, so for example here we are talking about a gene that codes for hemoglobin. Okay, I hope you know what is hemoglobin. Heme plus globin is hemoglobin. Heme is a protein, sorry, heme is an iron, okay, globin is a protein, and that iron plus a protein, you call it as hemoglobin. Okay, so when the particular gene that codes for hemoglobin is affected, automatically the protein is not produced properly, and that results in the deformed structure called as sickle cell. The RBCs will be of the form of sickle cell, and since erythrocytes or red blood cells are affected, red blood cells are otherwise called as erythrocytes. Okay, this condition we call as sickle cell anemia. Anemia always indicates whenever red blood cells are affected, use the word anemia. Okay, so in that way, this is a good example of gene or point mutation. Before we learned one example, that is in aneuploidy, we learned Down's syndrome, otherwise called as trisomy 21. Like that, now we have this sickle cell anemia, which is a good example of gene or point mutation. Okay, so that's all with today's class and today's lesson. Okay, so 18th lesson comes to an end.